The study of human evolution, from the earliest forms of life to the emergence of modern Homo sapiens, is a central subject in both biology and anthropology. But before we continue, please make sure to subscribe and leave like for more sleepy science videos. It seeks to uncover the processes, adaptations, and events that transformed simple microscopic organisms into complex beings capable of thought, language, and culture. This topic is not only significant for understanding the biological mechanisms that shaped our species, but also for placing humanity within the broader context of life on Earth. By tracing our origins, we gain insight into the interconnectedness of all living things and the evolutionary forces that have acted over billions of years. The journey begins with the appearance of the first cell, the fundamental unit of life, and continues through major evolutionary transitions: the rise of multicellular organisms, the diversification of vertebrates, and the development of primates. It then focuses on the emergence of early hominids, the gradual refinement of physical and cognitive traits, and the eventual arrival of anatomically modern humans. Cultural evolution, technological progress, and the formation of complex societies mark the final stages of this long process. Studying this path allows us to understand the deep history behind our existence and the biological legacy we carry today. The origin of life on Earth remains one of the most profound and challenging questions in science. While the fossil record preserves evidence of life dating back more than 3.5 billion years, the processes that transformed non-living chemical compounds into self-replicating, metabolically active systems are still not completely understood. Several leading hypotheses attempt to explain how life could have arisen under the physical and chemical conditions of the early Earth, and each provides a different perspective on the first steps toward biology. Among these are the primordial soup model, the hydrothermal vent theory, and the panspermia hypothesis. The primordial soup model, first popularized by Alexander Oparin and J.B.S. Haldane in the 1920s, proposes that life began in a soup of organic molecules formed in Earth's early oceans. According to this idea, the young Earth had a reducing atmosphere, rich in gases such as methane, CH4, ammonia, NH3, hydrogen, H2, and water vapor, H2O, but lacking significant amounts of oxygen, O2. This environment, combined with abundant energy sources like lightning strikes, ultraviolet radiation from the sun, and geothermal heat, could have driven the synthesis of simple organic compounds from inorganic precursors. These compounds, including amino acids, nucleotides, and simple sugars, may have accumulated in shallow ponds, tidal pools, or other water bodies, gradually forming increasingly complex molecules. The famous Miller-Urey experiment of 1953 provided experimental support for this model, demonstrating that amino acids could indeed be formed under simulated early Earth conditions using a mixture of gases, water, and electrical sparks. However, critics point out that the exact composition of the early atmosphere remains uncertain, and later studies suggest it may not have been as strongly reducing as Oparin and Haldane envisioned. An alternative view is offered by the hydrothermal vent theory. This hypothesis suggests that life may have originated at the bottom of the ocean, near hydrothermal vents, fractures in the Earth's crust that release superheated, mineral-rich water. These vents provide a stable supply of chemical energy through the release of hydrogen sulfide, methane, and other compounds, creating a chemically rich environment capable of supporting complex chemistry. The structures formed by vent minerals, particularly those associated with alkaline hydrothermal vents, contain microscopic pores that could have served as natural reaction chambers, concentrating molecules and facilitating the formation of more complex compounds. Additionally, The temperature gradients and pH differences found around these vents may have provided the energy needed for primitive metabolic reactions. One advantage of this scenario is that it protects nascent life from harmful ultraviolet radiation, which would have been intense on the surface before the formation of the ozone layer. Modern vent ecosystems, teeming with life that relies on chemosynthesis rather than photosynthesis, are often cited as analogs to early life-supporting environments. A third hypothesis, panspermia, shifts the focus away from Earth entirely. It proposes that life, or at least its chemical precursors, may have originated elsewhere in the universe and were transported to Earth by comets, asteroids, or interplanetary dust. This idea does not explain the ultimate origin of life, but it suggests that life may be a cosmic phenomenon rather than one unique to Earth. Evidence supporting panspermia includes the discovery of organic molecules, including amino acids, in meteorites such as the Murchison meteorite, as well as the resilience of certain microorganisms under extreme conditions, including the vacuum of space and high levels of radiation. While panspermia remains speculative and faces the challenge of explaining survival during atmospheric entry, it expands the scope of the search for life beyond our planet. To evaluate these hypotheses, it is important to consider the environmental conditions of early Earth. The planet formed about 4.54 billion years ago from the dust and gas left over after the formation of the Sun. In its earliest stages, Earth was a hot, molten world, frequently bombarded by asteroids and comets. As it cooled, a solid crust formed, and water began to accumulate on the surface, likely from both volcanic outgassing and impacts from icy bodies. The early atmosphere is thought to have been dense with carbon dioxide, CO2, nitrogen, N2, and water vapor, with smaller amounts of methane, ammonia, and hydrogen. The absence of free oxygen meant that ultraviolet radiation from the sun reached the surface unfiltered, providing both a hazard to fragile molecules and a potential energy source for chemical reactions. Volcanic activity during this time was intense, releasing gases and minerals that may have played key roles in prebiotic chemistry. Lightning strikes were common due to frequent storms, further contributing to the energy input needed for molecular synthesis. In addition, the proximity of the Moon, then much closer to Earth, created strong tidal forces, which could have caused regular cycles of concentration and dilution in shallow waters, potentially aiding the assembly of complex molecules. The chemical building blocks of life, amino acids, nucleotides, lipids, could have formed through various pathways under these conditions. Laboratory experiments, inspired by the Miller-Urey work but using updated atmospheric models, have continued to demonstrate that a wide variety of organic compounds can be synthesized from simple gases with the aid of energy sources. Fatty acids, for instance, can spontaneously form vesicles in water, creating primitive cell-like structures that could encapsulate and protect other molecules. These protocells may have been a crucial step toward the emergence of life, as they provide a controlled microenvironment for chemical reactions and allow for the separation of internal chemistry from the external environment. A central question in origin of life research is whether genetic information or metabolism came first. The RNA world hypothesis proposes that ribonucleic acid, RNA, capable of both storing information and catalyzing chemical reactions, may have been the first self-replicating molecule. In this view, early life began with RNA molecules that could copy themselves, eventually leading to the evolution of DNA and proteins. Others argue for a metabolism-first approach, in which networks of chemical reactions, possibly catalyzed by minerals at hydrothermal vents, produced increasing complexity until genetic systems emerged. These perspectives are not mutually exclusive, and both genetic and metabolic processes could have developed in parallel. Regardless of which specific pathway was followed, the transition from chemistry to biology required several critical steps. The formation of organic monomers, their polymerization into larger macromolecules, the emergence of self-replication, and the development of cell-like compartments. Each of these steps could have been influenced by the unique conditions of early Earth, from its chemical composition to its energy landscape. It is also possible that life began in more than one location, and that early forms competed or exchanged genetic material, with only one lineage surviving to become the ancestor of all modern life. This idea aligns with the concept of a last universal common ancestor, LUCA, a hypothetical organism from which all known life descends. LUCA is thought to have lived around 3.5 to 3.8 billion years ago and possessed the basic molecular machinery found in all cells today, including genetic material, ribosomes for protein synthesis, and a cell membrane. Ultimately, the origin of life on Earth remains a multidisciplinary puzzle involving chemistry, geology, planetary science, and biology. Each hypothesis offers compelling explanations for certain aspects of the process, but also faces unanswered questions. Future discoveries, whether from studying Earth's oldest rocks, simulating early conditions in the laboratory, or exploring extraterrestrial environments such as Mars or the icy moons of the outer planets, may bring us closer to understanding how lifeless matter gave rise to the rich diversity of organisms we see today. The question is not only of scientific interest, it touches on humanity's deepest curiosity about our place in the cosmos and whether life might be a universal phenomenon. The emergence of the first cells marks one of the most important transitions in the history of life on Earth. It represents the step from self-replicating molecules and loose chemical systems to bounded, organized entities capable of maintaining internal stability, reproducing, and evolving. These earliest cellular forms are believed to have been prokaryotic in nature, simple, single-celled organisms lacking a membrane-bound nucleus and other complex organelles. Yet, despite their apparent simplicity, these primitive cells were astonishingly effective at surviving and adapting, setting the stage for the vast diversity of life that would follow over billions of years. Prokaryotic cells are defined by a number of structural characteristics that distinguish them from later, more complex eukaryotic cells. They are generally much smaller, with diameters in the range of 0.1 to 5 micrometers. Their genetic material consists of a single circular molecule of DNA located in a region called the nucleoid, rather than enclosed within a membrane-bound nucleus. In addition to DNA, many also contain small circular DNA molecules called plasmids, which can carry auxiliary genes beneficial under certain environmental conditions. Prokaryotic cells lack the compartmentalization of functions seen in eukaryotes. Instead, all cellular processes occur within the same general space, though specialized regions or structures may exist. One of the key features enabling the survival and evolution of these first cells was the development of a cell membrane, a selectively permeable barrier composed primarily of a lipid bilayer. This membrane played multiple roles. It protected the internal environment from harmful external factors, controlled the passage of nutrients and waste products, and allowed for the maintenance of distinct chemical conditions inside the cell. The ability to regulate internal conditions, a process known as homeostasis, is fundamental to all forms of life. Without such regulation, the fragile chemical reactions necessary for metabolism and replication could not proceed efficiently or reliably. The chemical composition of early cell membranes may have differed significantly from that of modern organisms. In the absence of complex biochemical pathways, primitive membranes may have been composed of simple fatty acids and related molecules that could form spontaneously in prebiotic conditions. Experiments have shown that these fatty acids can self-assemble into vesicles in water, creating enclosed compartments that resemble modern cell membranes in their basic function. Over time, evolutionary processes would have refined these structures, leading to more stable and versatile phospholipid membranes found in contemporary prokaryotes. Inside these first cells, life's essential chemical processes began to take shape. The genetic material, most likely in the form of RNA initially, and later DNA, stored the instructions for building and maintaining the cell. RNA's ability to act as both an information carrier and a catalyst makes it a strong candidate for the first genetic system. In the so-called RNA world hypothesis, early RNA molecules could both store genetic information and catalyze their own replication or assist in the formation of other essential molecules. As life evolved, DNA eventually replaced RNA as the primary genetic material due to its greater chemical stability, while RNA's catalytic roles were largely taken over by proteins, though some catalytic RNAs, known as ribozymes, remain in modern cells. The metabolic diversity of prokaryotes was, and still is, extraordinary. Even in their earliest forms, these cells likely exploited a wide range of chemical energy sources available in their environment. Some may have been heterotrophic, relying on organic molecules produced by non-living chemical processes or other organisms. Others could have been autotrophic, synthesizing organic compounds from inorganic sources. Among the autotrophs, different metabolic pathways evolved. Photosynthesis, which uses light energy to produce sugars, and chemosynthesis, which derives energy from the oxidation of inorganic substances such as hydrogen sulfide or iron compounds. In early Earth's oxygen-free atmosphere, photosynthesis would have been anoxygenic, meaning it did not produce oxygen as a byproduct, relying instead on electron donors like hydrogen sulfide. Oxygen-producing photosynthesis, most famously carried out by cyanobacteria, would appear later and dramatically alter the planet's atmosphere and the trajectory of evolution. The evolution of metabolic pathways required enzymes, protein catalysts that accelerate chemical reactions. The earliest enzymes may have been small peptides or ribozymes, with greater complexity and efficiency developing over time. Enzymatic control over chemical reactions allowed cells to extract more energy from available resources and channel it into growth, replication, and maintenance. Reproduction in these early prokaryotic cells was likely through simple binary fission, a process in which the cell grows, replicates its genetic material, and then divides into two daughter cells. This method is efficient, allowing for large populations and high mutation rates, which in turn fuel evolutionary change. The absence of sexual reproduction in early life did not prevent genetic exchange entirely. Horizontal gene transfer, a process in which genetic material moves between cells without reproduction, may have been common, enabling rapid sharing of advantageous traits. This exchange of genetic material could have occurred through direct contact, uptake of free DNA from the environment, or viral intermediates. The oldest fossil evidence for life on Earth comes in the form of stromatolites, layered sedimentary structures formed by the activity of microbial communities, particularly cyanobacteria. These structures date back at least 3.5 billion years, indicating that complex, organized prokaryotic ecosystems existed relatively soon after the planet's surface became hospitable. Chemical signatures of ancient life, such as isotopic ratios of carbon indicative of biological activity, push the timeline of life's origin potentially even earlier, to over 3.8 billion years ago. The first cells faced numerous challenges in their environments. Ultraviolet radiation from the sun was intense in the absence of an ozone layer, potentially damaging genetic material and other cellular components. Fluctuations in temperature, pH, and salinity would have been common. Yet these conditions may also have driven adaptation, selecting for cells with protective mechanisms such as DNA repair enzymes, heat shock proteins, and robust membrane structures. Over time, certain lineages of prokaryotes adapted to extreme environments, hot springs, deep-sea vents, acidic pools, that are still inhabited today by extremophiles, organisms whose survival strategies likely mirror those of ancient life. The emergence of the first cells also set the foundation for the major evolutionary innovations to come. The existence of a stable, heritable genetic system allowed for the accumulation of beneficial mutations and the refinement of cellular processes. Metabolic diversity enabled life to colonize a wide range of environments and to exploit different energy sources. The compartmentalization provided by cell membranes facilitated more efficient and controlled biochemical reactions. From these humble beginnings, two major branches of prokaryotic life emerged, the bacteria and the archaea. While both share the general prokaryotic organization, they differ significantly in their genetic sequences, membrane chemistry, and certain aspects of metabolism. Archaea often thrive in extreme environments and have unique membrane lipids that make them more resilient to harsh conditions. The divergence of these two domains of life represents one of the earliest and most fundamental splits in the evolutionary tree, and it underscores the adaptability of prokaryotic forms. In the grand narrative of life on Earth, the appearance of the first cells was not a sudden event, but the result of countless small steps. Chemical innovations, structural refinements, and environmental challenges over vast spans of time. Once established, cellular life proved remarkably persistent, weathering planetary changes, asteroid impacts, and climatic shifts. Even today, prokaryotes remain the most numerous and diverse organisms on the planet, inhabiting every conceivable environment and playing critical roles in global ecosystems. The legacy of these early cells is universal. Every living organism, from bacteria to humans, carries the imprint of their basic design, a cell enclosed by a membrane, containing genetic material and powered by metabolic processes. The biochemical pathways that sustain life today, glycolysis, protein synthesis, DNA replication, trace their origins to this ancient era. Understanding the emergence of the first cells not only illuminates the deep history of life, but also informs our search for life beyond Earth, guiding scientists in identifying the conditions and markers that might indicate biological activity on other worlds. The emergence of eukaryotic cells was one of the most significant evolutionary transitions in the history of life, marking a profound leap in cellular complexity and biological potential. Eukaryotic cells, characterized by the presence of a membrane-bound nucleus and numerous specialized organelles, differ sharply from their prokaryotic ancestors. This transformation did not occur abruptly. Rather, it was the culmination of billions of years of evolutionary experimentation, involving intricate changes in cell structure, metabolism, and genetic organization. The prevailing explanation for the origin of key eukaryotic features, particularly mitochondria and chloroplasts, is the endosymbiotic theory, a model that describes how ancient prokaryotes entered into mutually beneficial relationships, eventually becoming permanent components of a single, integrated cell. At the heart of the eukaryotic cell lies the nucleus, a double membrane-bound compartment that houses the cell's genetic material in the form of linear chromosomes. This compartmentalization protects DNA from potentially damaging reactions in the cytoplasm, allows for more sophisticated regulation of gene expression, and supports the maintenance of larger and more complex genomes. The evolution of the nucleus likely involved the invagination of the plasma membrane in an ancestral prokaryote, forming an internal compartment around the genetic material. This step alone was revolutionary, as it enabled a greater separation between transcription, the production of RNA from DNA, and translation, the synthesis of proteins from RNA, leading to more precise control over cellular processes. The endosymbiotic theory provides the most compelling explanation for the origin of mitochondria and chloroplasts, two organelles central to eukaryotic energy metabolism. According to this theory, a large ancestral cell, most likely an archaeal-type prokaryote, engulfed smaller bacteria capable of aerobic respiration. Instead of digesting them, the host and the engulfed cells formed a symbiotic relationship. The bacteria provided the host with a more efficient means of generating energy using oxygen, while the host offered the bacteria a stable environment and access to nutrients. Over time, the engulfed bacteria became mitochondria, losing much of their original genome as genes were either discarded or transferred to the host's nuclear DNA. Mitochondria retain their own small circular DNA and reproduce independently of the host cell by a process similar to binary fission, a clear vestige of their bacterial ancestry. The presence of double membranes around mitochondria supports the engulfment scenario, with the inner membrane corresponding to the original bacterial membrane and the outer membrane derived from the host's engulfing vesicle. The acquisition of mitochondria was a pivotal moment in evolution. Aerobic respiration produces far more ATP, the universal energy currency of life, than anaerobic processes, allowing cells to power more demanding biochemical pathways and to grow larger and more complex. A parallel process explains the origin of chloroplasts in photosynthetic eukaryotes, such as plants and algae. In this case, the host cell engulfed a photosynthetic cyanobacterium, which retained its ability to capture light energy and convert it into chemical energy through photosynthesis. Like mitochondria, chloroplasts possess their own DNA, ribosomes, and double membranes, consistent with their descent from free-living bacteria. This secondary endosymbiosis granted the host cell the ability to produce its own organic molecules directly from sunlight, dramatically expanding ecological opportunities. The rise of eukaryotic cells involved not only the acquisition of energy-producing organelles, but also the development of a highly organized internal architecture. The endomembrane system, comprising the endoplasmic reticulum, Golgi apparatus, lysosomes, and various transport vesicles, facilitates the synthesis, modification, sorting, and transport of proteins and lipids. The rough studded with ribosomes, is a hub for protein synthesis, while the smooth is involved in lipid production and detoxification. The Golgi apparatus acts as a central packaging and distribution center, modifying proteins and directing them to their proper destinations. Lysosomes, containing digestive enzymes, break down waste materials and recycle cellular components. This compartmentalization of functions not only increases efficiency, but also prevents interference between incompatible biochemical reactions. The cytoskeleton, a dynamic network of protein filaments, provides structural support, maintains cell shape, and enables movement both within the cell and of the cell itself. Microtubules, actin filaments, and intermediate filaments each play distinct roles. Microtubules serve as tracks for organelle transport and are essential for chromosome segregation during cell division. Actin filaments support cell motility and shape changes. Intermediate filaments provide mechanical stability. The cytoskeleton's versatility allowed eukaryotic cells to adopt a wide range of shapes, to engage in active predation through processes such as phagocytosis, and eventually to form complex multicellular structures. One of the major advantages of the eukaryotic cell design is the capacity to maintain and regulate a much larger genome. This increase in genetic material provided the raw information needed to code for more proteins, more regulatory elements, and more complex interactions between cellular systems. Gene duplication events, facilitated by the greater genomic capacity, allowed for functional diversification, where one copy of a gene could retain its original role while the other evolved a new function. Over evolutionary time, this led to the emergence of entirely new metabolic pathways, structural proteins, and signaling networks. The appearance of eukaryotes fundamentally altered the evolutionary landscape. They could grow larger than prokaryotes, both in terms of individual cell size and in the complexity of multicellular assemblies. This was made possible in part by their energy surplus from mitochondria and, in photosynthetic lineages, from chloroplasts. Larger cell size, however, also brought challenges, including slower diffusion rates for nutrients and waste products. The development of active transport systems and intracellular trafficking pathways helped overcome these limitations. The earliest eukaryotic cells likely arose more than 1.6 to 2.1 billion years ago, as suggested by fossil evidence and molecular clock analyses. Microfossils from this period show larger cell sizes and complex morphologies consistent with eukaryotic features. In addition, certain biomarkers, molecular fossils such as steranes derived from sterols, which are synthesized only by eukaryotes, appear in rocks of similar age. The origin of eukaryotes appears to have been a singular event in life's history, though some evidence suggests that secondary and even tertiary endosymbiotic events occurred later in the evolution of certain algae and protists. Eukaryotic cells also laid the groundwork for the eventual rise of multicellularity. Their internal complexity allowed for specialized cell types to evolve, each performing distinct functions within a larger organism. Communication between cells via signaling molecules and direct physical connections became possible, enabling coordinated growth, development, and adaptation. The flexibility of the eukaryotic genetic and structural toolkit was critical to the diversification of life into the countless forms we see today. In addition to mitochondria and chloroplasts, many eukaryotic cells possess other membrane-bound organelles that further increase their versatility. Peroxisomes, for instance, are involved in breaking down fatty acids and detoxifying harmful substances. Vacuoles serve as storage compartments and help maintain osmotic balance, particularly in plant cells. The diversity of organelles reflects the ability of eukaryotic cells to compartmentalize and specialize, each compartment functioning as a miniature factory with a specific role in maintaining the life of the cell. From an evolutionary perspective, the origin of eukaryotes was a profound innovation because it merged distinct life forms into a single cooperative unit. The endosymbiotic relationships that produced mitochondria and chloroplasts were not simply one-time acquisitions. They required a long period of integration, during which genetic material was exchanged, metabolic pathways were coordinated, and mutual dependence deepened. Over time, the symbionts lost the ability to live independently, becoming permanent residents of the host cell. This transformation illustrates a recurring theme in evolution. Cooperation between different organisms can lead to entirely new levels of complexity and opportunity. Today, every plant, animal, fungus, and protist descends from that ancient lineage of eukaryotic cells. Their success lies in the combination of energy efficiency, organizational complexity, and genetic capacity, all of which originated with the innovations of compartmentalization and endosymbiosis. By understanding the origin of eukaryotic cells, scientists not only gain insight into a pivotal chapter of life's history, but also develop a deeper appreciation for the intricate partnerships that can shape the course of evolution. The leap from simple prokaryotic cells to the complex, dynamic architecture of eukaryotes was one of the most transformative events on Earth, setting the stage for the explosion of diversity that would follow in the ages to come. The evolution of multicellularity represents one of the most important and transformative events in the history of life on Earth. It marks the transition from simple aggregates of single-celled organisms, each functioning largely independently, to fully integrated, cooperative entities composed of many specialized cells working together toward the survival and reproduction of the whole organism. This shift allowed for an unprecedented increase in complexity, size, and ecological diversity. It also laid the foundation for the development of tissues, organs, and ultimately the vast array of plant and animal life that dominates the planet today. Multicellularity did not emerge in a single, linear event, but arose multiple times independently across different lineages, including animals, plants, fungi, algae, and certain bacteria. This convergence suggests that the evolutionary pressures and advantages of living as a coordinated multicellular unit were significant and recurring throughout life's history. The earliest forms of multicellular life were likely loose colonies of genetically identical cells that remained together after cell division. In such colonies, cells might have initially benefited simply from being in close proximity, perhaps for more efficient feeding or protection from environmental stresses. One of the critical steps toward true multicellularity was the evolution of mechanisms for cell adhesion. In unicellular colonies, cells could come together temporarily or remain loosely connected, but stable multicellular structures required specialized molecules that allowed cells to stick together reliably. In animals, these are adhesion proteins such as cadherins and integrins. In plants, pectins and other cell wall components serve a similar role. These adhesion systems not only held cells together physically, but also created a framework for communication and coordination among them. Communication between cells was another essential innovation. Multicellular organisms need to coordinate the activities of their various cells so that the organism can grow, respond to its environment, and repair itself. This coordination is achieved through chemical signaling molecules, such as hormones in animals or phytohormones in plants, as well as through direct physical connections between cells, like gap junctions in animals or plasmidesmata in plants. Such communication systems allowed cells to exchange information about their state, needs, and environmental conditions, enabling the organism to act as an integrated whole rather than as a mere collection of individual cells. Once stable adhesion and communication were in place, natural selection could favor the emergence of cell differentiation, the process by which cells take on specialized structures and functions. In a single-celled organism, each cell must perform all of life's essential processes, from energy production and reproduction to environmental sensing and defense. In a multicellular organism, different cells can become specialized for particular tasks, such as movement, nutrient absorption, reproduction, or structural support. This division of labor greatly increases efficiency and allows for the development of tissues, where groups of similar cells work together to perform specific functions. The formation of tissues was a crucial milestone in multicellularity. In animals, for example, the four basic tissue types, epithelial, connective, muscle, and nervous tissue, provide structural support, movement, communication, and defense. In plants, tissues are specialized for photosynthesis, nutrient transport, growth, and reproduction. Each tissue type is composed of cells with distinct shapes, structures, and molecular machinery optimized for their particular role. This organization enables multicellular organisms to perform complex functions that no single-celled organism could manage. Multicellularity also offered new ecological opportunities and survival advantages. Larger size, made possible by the coordinated growth of many cells, provided protection from predators and allowed organisms to access new resources. Large multicellular organisms could move through their environments in new ways, capture larger prey, or reach sunlight more effectively in crowded ecosystems. Additionally, the ability to regenerate damaged tissues and replace cells allowed multicellular life to withstand injuries that would be fatal to a single-celled organism. However, multicellularity also introduced new challenges. Cells within an organism had to balance their own survival with the survival of the whole organism, creating the potential for conflict. A cell that grew or reproduced uncontrollably could harm the organism, as seen in cancer. To prevent such problems, multicellular organisms evolved complex regulatory systems to control cell division, differentiation, and death. Programmed cell death, or apoptosis, became an important mechanism for removing damaged or unnecessary cells, maintaining the health and stability of the organism. The fossil record provides glimpses into the early history of multicellular life. Simple multicellular forms, such as filamentous algae, appear in rocks more than 1 billion years old. By around 600 million years ago, during the late Precambrian, the first large, complex multicellular organisms emerged, as evidenced by the Ediacaran biota, soft-bodied organisms with a variety of shapes and sizes. Shortly thereafter, the Cambrian explosion around 541 million years ago saw a rapid diversification of multicellular life, leading to the appearance of most major animal groups. The evolutionary routes to multicellularity varied among different lineages. In plants and fungi, multicellularity arose from ancestors that already had rigid cell walls, influencing how their cells could interact and specialize. In animals, the absence of rigid cell walls allowed for greater flexibility in movement and shape, contributing to the evolution of muscle and nerve tissues. Some lineages, such as certain algae and slime molds, display intermediate stages of multicellularity, with organisms that can switch between unicellular and multicellular forms depending on environmental conditions. These transitional forms offer valuable insights into how multicellular life could have evolved from unicellular ancestors. The genetic basis of multicellularity involves both the modification of existing genes and the evolution of entirely new ones. Many of the genes involved in cell adhesion, communication, and differentiation existed in unicellular organisms, but served different functions, such as recognizing environmental cues or attaching to surfaces. In multicellular lineages, these genes were co-opted and modified for new roles in maintaining the structure and function of the organism. Gene duplication provided additional raw material for evolution, allowing one copy of a gene to take on a specialized role while the other maintained its original function. Multicellularity can be considered both a product of cooperation and a triumph over the potential for internal conflict. The cells of a multicellular organism are genetically identical or nearly so, meaning they share the same evolutionary interests, the survival and reproduction of the organism as a whole. This genetic unity makes cooperation stable, as each cell benefits from contributing to the organism's success. However, mutations can disrupt this balance, creating selfish cells that prioritize their own replication over the organism's well-being. The evolution of regulatory systems to suppress such behavior was essential for the long-term stability of multicellular life. In modern ecosystems, multicellular organisms dominate in terms of visible biomass and complexity, from towering redwood trees to blue whales. Yet single-celled life remains abundant and ecologically vital, occupying niches that multicellular organisms cannot. The coexistence of unicellular and multicellular life reflects the fact that both strategies have advantages and disadvantages depending on environmental conditions. Multicellularity opened new possibilities for complexity, behavior, and interaction, but it came at the cost of greater energy demands and the need for elaborate coordination systems. In summary, the shift from single-celled colonies to fully integrated multicellular organisms was driven by the evolution of cell adhesion, communication, and differentiation, enabling the formation of tissues and the division of labor. This cooperation increased survival potential, allowed access to new ecological niches, and laid the groundwork for the remarkable diversity of life that followed. Multicellularity was not an inevitable outcome, but rather one of several possible evolutionary strategies, and its repeated emergence in different lineages highlights its profound adaptive value. From the earliest cell clusters to the complex bodies of plants and animals today, multicellularity stands as a defining feature of life's evolutionary narrative, illustrating how cooperation and specialization can transform the very architecture of living systems. The Cambrian explosion, occurring approximately 541 million years ago, represents one of the most remarkable and transformative periods in the history of life on Earth. Within a geologically brief span of roughly 20 to 25 million years, the diversity and complexity of organisms increased dramatically, leading to the appearance of most of the major animal phyla that exist today. This event marked a decisive shift from the relatively simple, soft-bodied life forms of the Precambrian to a world populated by organisms with hard shells, complex body plans, advanced sensory organs, and active modes of movement. The Cambrian explosion fundamentally reshaped Earth's biosphere, setting the stage for the ecological structures and interactions we recognize in modern ecosystems. Before the Cambrian, life on Earth was dominated by microbial mats, single-celled organisms, and a limited range of multicellular soft-bodied animals, such as those preserved in the Ediacaran biota. These organisms often exhibited simple radial or frond-like body structures, and many lacked clear evidence of sensory systems, specialized appendages, or skeletons. While there was some diversity, it was modest compared to what emerged in the Cambrian period. Then, in a relatively short time, the fossil record reveals a burst of morphological innovation and ecological diversification. One of the defining features of the Cambrian explosion was the emergence of complex body structures, often organized according to the principles of bilateral symmetry. Bilaterians, organisms with a left and right side, a front and back, and specialized head and tail regions, appeared in abundance. This symmetry was accompanied by the development of centralized nervous systems, enabling more sophisticated sensory processing and coordinated movement. For the first time, animals had the capability to actively hunt, avoid predators, and interact with their environments in dynamic ways. Another major innovation of the Cambrian period was the development of hard parts, shells, exoskeletons, spicules, and mineralized plates. This provided structural support, facilitated muscle attachment, and offered protection against predators. The appearance of these defensive structures has been interpreted as part of an evolutionary arms race, in which predators and prey co-evolved new adaptations to outcompete each other. Trilobites, for example, became one of the most iconic and diverse groups of Cambrian animals, protected by their articulated exoskeletons and capable of burrowing or swimming to escape danger. Brachiopods, mollusks, and early echinoderms also evolved mineralized coverings, contributing to the diversity of fossilizable remains from this period. Specialized sensory organs also became more common and sophisticated during the Cambrian explosion. Many organisms developed compound eyes, light-sensitive patches, and antennae, enabling them to detect movement light, and chemical cues with greater precision. The fossil record from sites such as the Burgess Shale in Canada reveals exquisitely preserved eyes in arthropods like Anomalocaris, suggesting that visual predation became an important ecological driver. The ability to see and be seen likely intensified evolutionary pressures, promoting the development of camouflage, armor, and complex behaviors. The causes of the Cambrian explosion have been the subject of intense scientific debate, and multiple factors likely acted together to trigger this rapid diversification. One leading hypothesis focuses on changes in environmental conditions, particularly the increase in atmospheric and oceanic oxygen levels. Higher oxygen concentrations would have supported larger body sizes, more active metabolisms, and the development of energetically costly features such as muscles and nervous systems. Geochemical evidence indicates that oxygen levels rose significantly in the late Precambrian, potentially reaching a threshold that made these complex adaptations feasible. Another possible factor was the breakup of supercontinents and the resulting changes in ocean circulation and nutrient distribution. The erosion of newly exposed continental areas could have delivered increased amounts of essential minerals and nutrients into the oceans, fueling primary productivity and expanding the base of the food web. A richer and more stable energy supply could have supported larger populations and more complex trophic interactions, paving the way for ecological diversification. Ecological interactions themselves may have been a powerful driver of evolutionary change. The emergence of active predators in the Cambrian created strong selective pressures on prey species to evolve defenses such as hard shells, burrowing behavior, spines, and toxins. This, in turn, would have driven predators to develop more effective hunting strategies, stronger appendages, and more advanced sensory systems. Such predator-prey dynamics could produce a feedback loop of innovation, with each side continually escalating its adaptations in response to the other. Developmental biology offers another perspective on the Cambrian explosion. Advances in the genetic and molecular mechanisms that control body patterning, particularly the evolution and diversification of Hox genes, may have provided the developmental flexibility needed to produce a wide variety of new body plans. Hox genes are responsible for determining the identity of body segments along the head-to-tail axis, and small changes in their function or expression can result in significant morphological differences. The co-option of existing genetic pathways for new purposes could have enabled rapid experimentation with novel forms and structures during the Cambrian period. It is important to note that while the Cambrian explosion appears sudden in the fossil record, many of the lineages that flourished during this time likely had Precambrian origins. Molecular clock estimates suggest that the ancestors of several major animal groups diverged tens of millions of years before their first clear fossil appearances. Thus, the explosion may reflect a combination of evolutionary innovations, ecological opportunities, and improved preservation of hard-bodied organisms rather than an instantaneous burst of origination. Nonetheless, the Cambrian period marks the point at which these lineages became ecologically prominent and morphologically diverse. Fossil sites from the Cambrian period provide invaluable insights into this evolutionary event. The Burgess Shale in Canada, the Chengjiang biota in China, and the Sirius Posset Formation in Greenland have yielded exceptionally well-preserved fossils of both hard and soft-bodied organisms, allowing scientists to reconstruct the anatomy, lifestyles, and ecological roles of a wide range of species. These sites reveal not only familiar groups like arthropods, sponges, and mollusks, but also many weird wonders that do not fit neatly into modern classifications, such as Opabinia with its 5 eyes and proboscis, or Hallucigenia with its spines and tentacles. Such forms highlight the experimental nature of early animal evolution, when natural selection explored a variety of body plans, some of which left no direct descendants. The Cambrian explosion also had profound long-term consequences for life on Earth. The establishment of complex food webs with multiple trophic levels created more stable and resilient ecosystems. The diversification of body plans provided the raw material for future evolutionary radiations, including the colonization of land by plants and animals hundreds of millions of years later. Many of the fundamental anatomical architectures seen today, such as the segmented bodies of arthropods, the notochord of chordates, and the radial symmetry of echinoderms, were firmly established during the Cambrian and have persisted through subsequent eras. In summary, the Cambrian explosion was a pivotal event in the history of life, characterized by the rapid diversification of animal forms, the emergence of hard parts and advanced sensory systems, and the establishment of complex ecological interactions. Its causes likely included a combination of environmental changes, ecological dynamics, and developmental innovations that together created the conditions for a burst of evolutionary creativity. While its apparent suddenness may be partly an artifact of the fossil record, the Cambrian period nonetheless marks a real and dramatic transformation in the biosphere, one whose legacy continues to shape life on Earth today. The colonization of land by living organisms marks one of the most transformative chapters in the history of life on Earth, fundamentally altering ecosystems, atmospheric composition, and evolutionary trajectories. For billions of years after the origin of life, Earth's biosphere was dominated by aquatic organisms, from microscopic plankton to large marine animals. Oceans, lakes, and rivers offered abundant water, stable temperatures, and a medium in which nutrients and gametes could disperse easily. Life's gradual transition from water to land was neither sudden nor simple. It was a process that unfolded over hundreds of millions of years and required a suite of evolutionary innovations to overcome the challenges of a terrestrial environment. Both plants and animals had to solve similar problems: gravity, desiccation, reproduction outside of water, and mobility, although they addressed these challenges in different ways. The earliest pioneers of land were likely simple photosynthetic organisms similar to modern algae, which may have periodically inhabited moist shorelines and tidal flats. These early colonizers faced the problem of water loss due to evaporation, which is far more pronounced in air than in water. To survive, they needed protective structures to minimize desiccation. This adaptation took the form of the cuticle, a waxy, hydrophobic layer covering the surface of stems and leaves, reducing water loss while still allowing light to penetrate for photosynthesis. The development of a cuticle was a critical first step in making a permanent life on land viable for plants. However, a cuticle alone was not enough. Terrestrial plants also needed a way to transport water and dissolved minerals from the soil to aerial tissues and to distribute sugars produced in leaves to other parts of the organism. This led to the evolution of vascular tissue, xylem for conducting water and minerals upward from the roots, and phloem for transporting sugars and other organic compounds throughout the plant. Vascular systems allowed plants to grow taller, capturing more sunlight and outcompeting shorter vegetation. They also enabled colonization of areas farther from bodies of water, as vascular tissue could maintain hydration deep within the plant structure. Roots were another revolutionary adaptation. While early land plants may have anchored themselves with simple rhizoids, true roots provided both stability in the substrate and an efficient interface for absorbing water and nutrients. Roots also formed symbiotic relationships with fungi, mycorrhizae, vastly improving nutrient uptake, particularly phosphorus, thereby enhancing plant survival and growth in challenging soils. Reproduction posed a particularly complex challenge for plants transitioning to land. In aquatic environments, gametes can disperse freely in the water, but on land, desiccation and lack of a liquid medium threaten fertilization. Early land plants, such as bryophytes, still relied on water for reproduction, producing motile sperm that swam to eggs during wet periods. Later plants evolved pollen grains, microscopic structures containing the male gametophyte protected by a tough outer wall of sporopollenin, which allowed fertilization to occur without freestanding water. Seeds represented an even greater innovation, enclosing the developing embryo in a protective coat with a nutrient supply, enabling plants to spread to drier and more variable habitats. While plants were gradually securing their place on land, animals were also making their first ventures out of aquatic environments. The earliest terrestrial animals were likely arthropods, including primitive millipede-like myriapods and scorpions, which may have fed on detritus or early plants along moist shorelines. These animals already had exoskeletons, a key advantage in preventing water loss and providing structural support against gravity. Additionally, the segmentation and jointed appendages of arthropods offered versatile locomotion on uneven terrain. For vertebrates, the transition to land was more complex. The ancestors of tetrapods were lobe-finned fish that lived in shallow waters, such as Tiktaalik, which displayed both fish-like features, gills, fins, and tetrapod-like adaptations, strong limb bones, a neck, and lungs. In aquatic settings subject to periodic drying, the ability to breathe air became a vital survival trait. Many early fish had lungs in addition to gills, derived from outpocketings of the gut. These primitive lungs allowed them to gulp air when oxygen levels in water were low. Over evolutionary time, lungs became the primary respiratory organ for terrestrial vertebrates, supplemented in some groups by moist skin capable of gas exchange, as in amphibians. The shift from fins to limbs was equally crucial. Fins are effective in water, but terrestrial locomotion requires the ability to support the body's weight against gravity and to propel it forward on solid ground. The bones of the lobe-finned fish's fins gradually evolved into the tetrapod limb structure, humerus, radius, ulna, and corresponding bones in the hind limbs, capable of bearing weight and enabling walking or hopping. Amphibians, the first vertebrates to live partly on land, still retain ties to water for reproduction and often inhabited moist environments, but their limbs allowed them to exploit new niches beyond the shoreline. Another major challenge for terrestrial animals was desiccation resistance. Gills function only when moist, and exposed skin rapidly loses water in dry air. Many arthropods and reptiles evolved impermeable outer coverings, chitinous exoskeletons, or keratinized scales, to minimize water loss. For vertebrates, internal fertilization and the evolution of the amniotic egg represented a breakthrough. The amniotic egg, with its multiple protective membranes and a fluid-filled cavity, allowed embryos to develop on land without the risk of desiccation. This adaptation freed reptiles, birds, and later mammals from the reproductive dependence on aquatic environments. The colonization of land also had profound ecological effects. Plants were the first to establish a foothold, stabilizing soils and beginning the slow process of forming terrestrial ecosystems. As plant communities expanded, they provided both habitat and food sources for herbivorous arthropods, which in turn supported the emergence of terrestrial predators. Early forests, composed of tall vascular plants such as lycophytes and ferns, transformed atmospheric chemistry by increasing oxygen levels and reducing carbon dioxide, contributing to climatic shifts that favored further diversification of land-based life. The spread of plants and animals onto land was not uniform or immediate. It was influenced by climatic conditions, continental arrangements, and local geography. Coastal wetlands, river deltas, and floodplains likely served as key transitional zones where aquatic and terrestrial features overlapped. Organisms in these habitats were under strong selective pressure to adapt to fluctuating water levels, seasonal droughts, and new types of predators and competitors. Once established, terrestrial life diversified rapidly. Plants developed new growth forms, such as woody stems for structural support, and new reproductive strategies, such as flowers to attract animal pollinators. Animals diversified into a variety of forms adapted for running, climbing, burrowing, and flying. The mutual influences of plants and animals, herbivory driving plant defenses, pollination fostering coevolutionary relationships, created dynamic and interdependent ecosystems. By the late Paleozoic, terrestrial environments supported complex food webs, from towering seed ferns and conifers to large amphibians, early reptiles, and diverse arthropods. The foundations laid by the earliest colonizers of land not only reshaped life on Earth, but also set the stage for the rise of dinosaurs, mammals, and eventually humans. The adaptations developed during this transition, structural support, internal water regulation, air breathing, and independent reproduction, remain fundamental to life on land today. The story of life's colonization of land is, at its core, a story of innovation driven by necessity. The harsh challenges of the terrestrial world forced plants and animals to rethink the basic mechanics of survival, leading to some of the most important evolutionary breakthroughs in history. Without roots and vascular systems, lungs and limbs, cuticles and amniotic eggs, the diversity of life we see on continents today, from vast forests to complex animal communities, would never have been possible. The evolution of vertebrates is one of the most remarkable narratives in the history of life, illustrating how a single lineage diversified into a vast array of forms adapted to almost every environment on Earth. The story begins in the Cambrian period, over 500 million years ago, when the first primitive vertebrates emerged in the seas. These early forms belong to a broader group of chordates, animals characterized by possessing, at some stage in their development, a notochord, a flexible rod-like structure that provides support, as well as a dorsal nerve cord, pharyngeal slits, and a postanal tail. The notochord represented a key evolutionary step toward the vertebral column or backbone, which would come to define the vertebrate subphylum. The earliest vertebrates were jawless fish known as agnathans. They had cartilaginous skeletons, simple muscular systems, and gill slits for filter feeding. Although lacking paired fins and jaws limited their predatory capabilities, they established the fundamental vertebrate body plan: a head with sensory organs, a brain encased in cartilage, and a segmented musculature for swimming. Over time, natural selection favored more efficient feeding and locomotion strategies, setting the stage for the next great innovation, the jaw. Jaws first appeared in the Silurian period, around 430 million years ago, and are thought to have evolved from modified gill arches. This change revolutionized vertebrate feeding by allowing organisms not only to bite and grasp prey, but also to process a wider variety of foods. Early jawed fish, or gnathostomes, diversified rapidly into groups such as placoderms, armored fish with bony plates, and cartilaginous fish like sharks and rays. At the same time, paired fins became more prominent, improving stability and maneuverability in the water. Skeletal structures grew more robust, and the development of mineralized bone provided both protection and support. The next major transition occurred with the rise of the lobe-finned fish, or sarcopterygians. Unlike their ray-finned counterparts, lobe-finned fish possessed muscular, fleshy fins supported by a series of bones homologous to the limbs of terrestrial vertebrates. This anatomy made it possible for them to push along the bottom of shallow waters or even move between water bodies during droughts. Some, like Tiktaalik, exhibited intermediate traits between fish and tetrapods, such as functional necks, sturdy ribs for supporting the body in shallow water, and limb-like fins. The move from water to land required more than structural changes to fins. It also demanded adaptations in respiration, circulation, and sensory systems. Lungs, which had evolved in some fish as an accessory breathing organ, became essential for air breathing, while circulatory systems adapted to pump blood effectively in a gravity-dominated environment. The emergence of amphibians in the Devonian period marked the first fully terrestrial vertebrates. Early amphibians such as Ichthyostiga and Acanthostiga still had strong ties to water, relying on aquatic environments for reproduction and maintaining moist skin for respiration. Amphibians developed stronger limb bones, digits instead of fin rays, and more complex musculature to handle locomotion on land. However, their dependence on external water sources limited their ability to exploit drier environments, leaving an ecological opportunity for further adaptations. This opportunity was seized during the Carboniferous period with the rise of amniotes, a group that included reptiles, birds, and mammals. The defining feature of amniotes is the amniotic egg, which contains specialized membranes, the amnion, chorion, and allantois, that protect and nourish the embryo while preventing desiccation. This adaptation freed vertebrates from the need to reproduce in water and opened up vast new terrestrial habitats. Early reptiles developed tough, scaly skin to reduce water loss, and their lungs became more efficient for air breathing. Reptiles diversified into numerous forms, from sprawling, lizard-like species to the ancestors of crocodiles, dinosaurs, and pterosaurs. The reptilian lineage split into two major evolutionary branches. One branch, the synapsids, gave rise to mammals, while the other, the diapsids, produced most modern reptiles and birds. Early synapsids, often called mammal-like reptiles, displayed a mix of reptilian and mammalian traits, including differentiated teeth for specialized feeding and a more upright posture. Over millions of years, synapsids evolved more efficient metabolisms, higher activity levels, and more complex social behaviors. One of the most significant shifts in this lineage was the gradual move toward endothermy, or warm-bloodedness. Endothermy allows animals to maintain stable internal body temperatures regardless of environmental conditions, enabling them to inhabit a wider range of climates and remain active during both day and night. This adaptation required a higher metabolic rate, supported by improvements in cardiovascular and respiratory systems, as well as the development of insulation in the form of fur. Meanwhile, diapsids produced two key subgroups: lepidosaurs, lizards, snakes, and tuatara, and archosaurs, crocodiles, pterosaurs, and dinosaurs. Among the dinosaurs, one lineage evolved feathers and eventually gave rise to birds, another instance of warm-bloodedness evolving independently. Birds retained many reptilian features, such as scaly legs and egg-laying, but their adaptations for flight, including lightweight skeletons, powerful pectoral muscles, and highly efficient respiratory systems, made them one of the most successful vertebrate groups. Mammals, emerging fully during the Mesozoic era, brought together a combination of traits that distinguished them from their reptilian ancestors: endothermy, fur, live birth in most species, and highly specialized teeth and jaws. Their three middle ear bones, derived from reptilian jaw elements, improved hearing sensitivity. The mammalian brain expanded significantly, particularly the neocortex, allowing for more complex behaviors, problem-solving, and social interactions. Mammals diversified after the extinction of the dinosaurs, filling ecological niches from small insectivores to large grazers and apex predators. They adapted to every major habitat, from deep oceans, whales and dolphins, to arid deserts, camels, and frozen polar regions, polar bears. Throughout vertebrate evolution, certain innovations repeatedly opened the door to new ecological opportunities. The backbone provided structural support and protection for the nerve cord, enabling greater mobility and larger body sizes. Jaws allowed for more diverse feeding strategies, promoting predator-prey arms races and ecosystem complexity. The amniotic egg and, later, live birth freed animals from dependence on aquatic reproduction, enabling full terrestrial lifestyles. Endothermy and associated physiological changes expanded the range of habitable environments and increased activity levels. Vertebrate evolution is not a simple linear progression but a branching tree, with multiple lineages experimenting with different solutions to the challenges of survival. Some innovations arose multiple times independently, such as flight in pterosaurs, birds, and bats, or warm-bloodedness in birds and mammals. Others, like the development of limbs from fins, happened once and then served as the basis for countless adaptations. By the time humans appeared, vertebrates had already conquered the seas, skies, and continents. The evolutionary journey from the first jawless fish to modern mammals involved a series of transformative steps, each building upon the last. This history reveals the power of natural selection to shape life in response to environmental pressures, competition, and opportunity. The legacy of these changes is evident in the diversity of vertebrates today, from tiny hummingbirds to massive blue whales, all descendants of a common ancestor that swam in the ancient oceans over 500 million years ago. The emergence of primates represents a pivotal chapter in the evolutionary story, marking the origin of a lineage whose unique anatomical, sensory, and behavioral traits would eventually culminate in the human species. Primates first appeared more than 55 million years ago during the Paleocene-Eocene transition, a period of global warming and expanding tropical forests. These environmental conditions created a landscape rich in fruit, flowers, and insects, with dense canopies providing both resources and challenges. In such habitats, natural selection favored adaptations for life in the trees, arboreal lifestyles that demanded agility, precise movement, and enhanced sensory coordination. One of the defining characteristics of primates is the structure of their hands and feet. Unlike many other mammals, primates developed grasping extremities with opposable thumbs or big toes, allowing them to securely hold onto branches and manipulate objects. This adaptation enabled them to forage for food with precision, groom each other for social bonding, and eventually use tools. Flexible shoulder joints, capable of a wide range of motion, complemented their grasping abilities, making them adept climbers and leapers. Their limbs often had a high degree of mobility, and their digits were equipped with flat nails rather than claws, enhancing tactile sensitivity. Vision became the dominant sense in primates, a shift from the olfactory reliance seen in many other mammals. Forward-facing eyes provided overlapping fields of view, granting binocular vision and improved depth perception, an essential adaptation for navigating complex, three-dimensional forest environments. This enhancement was supported by an expansion of the visual processing areas in the brain, particularly the occipital lobes. Color vision evolved in many primate lineages, likely to aid in distinguishing ripe fruits and young leaves from surrounding foliage. While early nocturnal primates had larger eyes adapted for low light, diurnal species optimized for sharp daytime vision. Another hallmark of primates is their relatively large brain size compared to body mass, a feature associated with advanced problem-solving, memory, and social behaviors. Brain regions linked to sensory integration, motor control, and complex cognition became increasingly prominent. This neural expansion supported the coordination of hand-eye movements, the recognition of social hierarchies, and the development of intricate communication systems. Large brains also facilitated behavioral flexibility, allowing primates to exploit a variety of food sources and adapt to changing environments. The earliest primates were small, arboreal creatures resembling modern tree shrews, though more specialized for life among branches. Fossils such as Plesiodopus from the Paleocene and Notharctus from the Eocene reveal transitional forms with adaptations for grasping and leaping. These early primates gave rise to two main suborders, the strepsirrhines and the haplorhines. Strepsirrhines, often referred to as persimians, include lemurs, lorises, and galagos. They tend to retain more ancestral features, such as a greater reliance on smell and a smaller brain-to-body ratio than later primates. Many are nocturnal, with large eyes adapted for night vision, and they often have a reflective layer behind the retina, tapetum lucidum, to improve light capture. Strepsirrhines also exhibit a grooming claw on one digit and a tooth comb, a specialized arrangement of lower teeth used for grooming fur. Despite their more basal traits, they show remarkable diversity and adaptability, thriving in environments ranging from Madagascar's rainforests to African woodlands. The second major suborder, haplorhines, encompasses tarsiers, monkeys, and apes, and represents a significant evolutionary shift. Haplorhines generally have larger brains, greater dependence on vision, and more complex social behaviors. Tarsiers, small primates from Southeast Asia, are an intriguing intermediate form. They are nocturnal like many strepsirrhines but lack the tapetum lucidum, relying instead on enormous eyes for light capture. They are highly specialized leapers, feeding primarily on insects and small vertebrates. Haplorhines soon split into two major infraorders, the New World monkeys, platyrrhines, and the Old World monkeys and apes, catarrhines. New World monkeys reached South America, likely by rafting across the Atlantic on vegetation mats from Africa around 40 million years ago. They evolved distinctive features such as broad, outward-facing nostrils and, in many species, prehensile tails capable of grasping branches. Examples include capuchins, spider monkeys, and howler monkeys, each adapted to different ecological niches. Old World monkeys and apes, on the other hand, evolved in Africa and Asia, with narrow, downward-facing nostrils and generally no prehensile tails. Old World monkeys diversified into two primary groups, the Cercopithecines, which often inhabit savannas and have cheek pouches for food storage, and the Colobines, primarily arboreal leaf eaters with complex stomachs for digesting fibrous plant material. This diversification allowed Old World monkeys to occupy a wide range of habitats, from tropical forests to mountainous regions and dry grasslands. Apes, or hominoids, emerged from Old World monkey ancestors approximately 25 million years ago. They differ from monkeys in several key respects. They lack tails, have a more upright posture, possess larger brains relative to body size, and often show greater flexibility in the shoulder and hip joints. Early apes like Proconsul retained many arboreal traits, but also showed the beginnings of adaptations for more versatile movement. Over time, apes split into lesser apes, gibbons, and great apes, the latter including orangutans, gorillas, chimpanzees, bonobos, and humans. The arboreal lifestyle shaped primate social structures and behaviors. Living in trees meant balancing the need for safety from predators with access to food resources, often leading to complex group dynamics. Many primates live in social groups that require cooperation, communication, and conflict resolution. Vocalizations, facial expressions, and body language became sophisticated means of conveying information. Grooming emerged as a critical social activity, reinforcing bonds and alliances within groups. Dietary flexibility was another key factor in primate success. While some specialized in fruits, frugivores, others consumed leaves, folivores, insects, insectivores, or a combination of these. This adaptability allowed primates to thrive in diverse environments, from dense tropical forests to more open woodland and savanna habitats. As climate changes in the Miocene reduced forest cover in some regions, certain lineages adapted to more terrestrial lifestyles, setting the stage for later hominid evolution. Throughout their evolutionary history, primates have demonstrated an extraordinary ability to exploit their environment through a combination of physical adaptations, sensory enhancements, and behavioral flexibility. The interplay between arboreal demands, dietary opportunities, and social complexity drove the refinement of traits such as grasping hands, binocular vision, and large brains. These characteristics not only ensured survival in ancient tropical forests, but also laid the foundation for the emergence of hominins and, ultimately, modern humans. The story of primate evolution is thus one of gradual specialization for complex, multidimensional living spaces, followed by diversification into a range of ecological roles. From the small, nocturnal persimians to the highly intelligent great apes, each branch of the primate family tree reflects millions of years of adaptation and innovation. It is within this lineage, shaped by the interplay of environment, anatomy, and behavior, that the seeds of humanity were sown, making the emergence of primates one of the most critical steps in the evolutionary journey toward Homo sapiens. The early hominid stage of evolution marks a critical turning point in the story of humanity, as it is during this period that our lineage began to exhibit traits clearly separating it from other apes. At the center of this transformation stands Australopithecus, a genus of hominins that lived between approximately 4 and 2 million years ago in various parts of Africa. These species were neither fully ape-like nor fully human, but rather represented a mosaic of traits from both worlds. They occupied an evolutionary space that linked the last common ancestor shared with chimpanzees to the first members of the genus Homo. Their biology, behavior, and ecological adaptations offer profound insights into how and why humans became distinct. One of the most defining features of Australopithecus was bipedalism, the ability to walk habitually on two legs. This adaptation is perhaps the single most important hallmark of the human lineage. Fossil evidence, including the famous partial skeleton of Australopithecus afarensis nicknamed Lucy, reveals a pelvis that was shorter and broader than that of apes, reorienting the muscles needed for upright walking. The femur angled inward toward the knee, allowing weight to be balanced over a single supporting leg during locomotion. Additionally, the structure of the foot shows a strong, non-divergent big toe and arches that helped absorb shock while walking. These anatomical changes point to a gait that was efficient for covering open ground, even if not as fast or as smooth as that of modern humans. The reasons for the evolution of bipedalism remain the subject of debate. One prevailing idea is that climatic changes in Africa during the late Miocene and early Pliocene led to the expansion of open savannas at the expense of dense forests. Early hominids faced a more mosaic landscape of grasslands and scattered trees, making upright walking advantageous for traveling longer distances between food sources, scanning for predators, and possibly carrying food or tools. Another hypothesis suggests that bipedalism evolved to reduce heat stress by minimizing the body's exposure to direct sunlight during the hottest parts of the day. The truth may lie in a combination of ecological pressures and energetic efficiency. Despite their upright posture, Australopithecus species retained many features suited for climbing and arboreal activity. Their long arms, curved fingers, and powerful upper body muscles suggest that they still spent significant time in trees, whether for feeding, sleeping, or avoiding predators. This mixed locomotion pattern, terrestrial bipedalism combined with arboreal agility, illustrates their transitional nature. They were not yet committed ground dwellers like later humans, but had moved far from the fully arboreal lifestyle of earlier primates. The brain size of Australopithecus was modest, averaging around 400 to 550 cubic centimeters, only slightly larger than that of modern chimpanzees. However, subtle changes in skull shape and the organization of the brain case hint at gradual neurological developments. While they did not possess the large, complex brains of later hominins, they may have been capable of rudimentary problem-solving, basic tool use, and social behaviors more advanced than those of other apes. Their face projected forward, with a pronounced jaw and large teeth adapted for processing a variety of foods. The thick enamel on their molars and the robust nature of their jaws indicate a diet that included hard or fibrous plant material, such as roots, tubers, and nuts, along with fruits and possibly small amounts of meat or insects. Several species of Australopithecus have been identified, each adapted to different environments. Australopithecus anamensis, the earliest known species, lived about 4.2 to 3.9 million years ago and shows a blend of primitive ape-like traits with clear evidence of habitual bipedalism. Australopithecus afarensis, which lived between about 3.9 and 3 million years ago, is one of the best known and most studied species due to abundant fossil evidence from East Africa. Its famous Lucy skeleton, along with footprints preserved at Litoli in Tanzania, provides direct proof that these hominins walked upright on a regular basis. Australopithecus africanus, found in South Africa and dating from around 3 to 2 million years ago, displayed slightly more human-like facial features and may represent a descendant of afarensis. Other more specialized forms, such as Australopithecus garhi and Australopithecus setiba, suggest that this genus was diverse and adaptive. A. garhi, for instance, has been associated with animal bones showing cut marks, raising the possibility that it used simple stone tools to extract meat and marrow, behaviors once thought to be unique to later Homo species. A. setiba, dating to around 2 million years ago, exhibits a curious blend of traits found in both Australopithecus and early Homo, leading some researchers to consider it a possible transitional form to our own genus. Socially, Australopithecus likely lived in small to medium-sized groups, similar to many modern primates. Group living would have provided protection against predators, opportunities for cooperative foraging, and support in rearing offspring. Sexual dimorphism, the difference in size between males and females, was moderate to pronounced in some species, suggesting a social structure that may have included competition among males for mates, possibly with polygynous tendencies. However, the degree of sexual dimorphism varied, indicating that social systems might not have been uniform across all species. The significance of Australopithecus in human evolution lies not just in its physical traits, but in its position in the evolutionary timeline. These hominins bridge the gap between our ape-like ancestors and the emergence of the genus Homo. They show that bipedalism preceded large brain size, challenging earlier assumptions that intelligence drove the earliest stages of human evolution. Instead, upright walking came first, with brain expansion and advanced tool use developing later. This order of events reshaped scientific understanding of what it means to be human. Environmental adaptability was another key factor in the success of Australopithecus. They thrived in varied habitats, from woodlands to open savannas, demonstrating flexibility in both diet and locomotion. This adaptability would prove vital for their descendants, as climate fluctuations continued to reshape African ecosystems in the millennia to come. Such versatility in coping with shifting conditions may have been inherited by the first members of Homo, enabling them to spread into new environments beyond Africa. By about 2 million years ago, some Australopithecus species had either evolved into or given way to the earliest Homo species, such as Homo habilis. These successors carried forward the upright posture and bipedal gait perfected by their Australopithecine ancestors, but paired them with larger brains, more sophisticated tools, and eventually the capacity for symbolic thought. In this way, Australopithecus stands as both a culmination of earlier primate evolution and a foundation for the human lineage. The fossil record of Australopithecus continues to grow as new discoveries are made across Africa, each adding detail to our understanding of this pivotal genus. Their story underscores the fact that human evolution was not a straight line, but a branching tree, with multiple species experimenting with different ways of living and adapting. Some branches ended, while others gave rise to entirely new forms. Australopithecus may be gone, but its legacy is literally built into our bones, in the shape of our pelvis, the structure of our spine, and the mechanics of our stride. In sum, Australopithecus represents the crucial step when our ancestors committed to life on two legs, while still maintaining connections to their arboreal past. It shows how selective pressures in a changing African landscape forged a creature that could walk upright, use its hands for tasks beyond locomotion, and adapt to a wide range of environments. Without the evolutionary experiments embodied by Australopithecus, the path to Homo sapiens would never have been possible, making this genus one of the most important links in the chain of our origins. The emergence of Homo habilis and Homo erectus marks a profound turning point in human evolution, representing the transition from more primitive hominins, such as Australopithecus, to species that exhibited distinctly human-like behaviors and adaptations. These two species, spanning a period from roughly 2.4 million to 140,000 years ago, demonstrate critical advancements in brain size, tool-making, social organization, and environmental adaptability, setting the stage for the eventual rise of Homo sapiens. Homo habilis, often called the handyman, lived between about 2.4 and 1.6 million years ago in East and possibly Southern Africa. It is widely regarded as the earliest known species of the genus Homo. The species had a cranial capacity ranging from about 510 to 600 cubic centimeters, larger than that of Australopithecus, but still smaller than later humans. This increase in brain size is significant because it correlates with more complex cognitive abilities, particularly in tool manufacture and use. Fossil evidence links H. habilis to the Oldowan stone tool tradition, which involved producing simple yet effective implements by striking flakes from a stone core. These tools, sharp-edged flakes for cutting and heavier cores for pounding, allowed H. habilis to process a wider variety of foods, including meat, roots, and tubers. Access to animal protein, whether through scavenging or hunting small prey, likely contributed to the nutritional support needed for brain growth. Dietary flexibility was one of H. habilis' key survival strategies. Unlike their Australopithecine predecessors, they were less specialized in their feeding habits. While still consuming plant matter such as fruits, nuts, and seeds, their use of tools expanded their ability to exploit animal resources, possibly scavenging carcasses left by predators. This adaptability would have been especially advantageous in the shifting Pleistocene environments, where climatic fluctuations caused periodic changes in vegetation and animal populations. Socially, H. habilis may have lived in small groups that cooperated in foraging and shared food, behaviors that fostered social bonds and learning. Although direct evidence for social structure is limited, the development of tool-making likely required observation, imitation, and some form of communication. These early communities may have had rudimentary proto-language abilities, consisting of gestures, vocalizations, and facial expressions to convey basic information about resources and dangers. By around 1.9 million years ago, Homo erectus appeared, possibly evolving from H. habilis or a closely related ancestor. H. erectus represents a major evolutionary leap in terms of anatomy, behavior, and geographic range. Their cranial capacity averaged between 800 and 1,200 cubic centimeters, significantly larger than that of H. habilis, indicating a substantial increase in cognitive potential. The skulls of H. erectus were elongated with a pronounced brow ridge, and the face projected less than in earlier species, showing a gradual trend toward modern human features. One of the most striking physical changes in H. erectus was in body proportions. Fossils such as the famous Turkana boy from Kenya reveal a tall, long-legged build well-suited for endurance walking and running. This adaptation would have greatly enhanced their ability to travel long distances in search of food and water, as well as to follow migrating herds. Their skeletons were more robust than those of modern humans, indicating a physically demanding lifestyle, but their limb proportions were essentially modern, enabling efficient movement across open landscapes. Tool technology also advanced considerably with H. erectus. While early populations may have used Oldowan tools inherited from H. habilis, they soon developed the Acheulean industry, characterized by large, bifacially worked hand axes and cleavers. These tools required more planning and skill to produce, suggesting an improved capacity for abstract thought and manual dexterity. Acheulean tools were versatile, used for cutting meat, processing plant material, and woodworking, reflecting a broader and more sophisticated approach to resource exploitation. Perhaps the most transformative innovation associated with H. erectus was the mastery of fire. Evidence from sites such as Wonderwork Cave in South Africa suggests that they were controlling fire as early as 1 million years ago. Fire provided numerous advantages. It offered warmth in colder climates, protection from predators, and the ability to cook food. Cooking not only made meat and starchy plants easier to chew and digest, but also increased the caloric availability of these foods, supporting further brain growth. Fire also created a social focal point, where group members could gather, strengthening bonds, and potentially fostering the exchange of knowledge. The geographic expansion of H. erectus is another hallmark of the species. They were the first hominins to leave Africa, dispersing into Eurasia and Southeast Asia. Fossil remains have been found in locations as far-flung as Georgia, Manasai, Indonesia, Java man, and China, Peking man. This wide distribution demonstrates their adaptability to diverse environments, from tropical forests to temperate grasslands, and even more arid regions. Their ability to migrate and settle in new territories likely relied on their improved physical endurance, tool-making skills, and possibly fire use. Social complexity also appears to have increased with H. erectus. While direct evidence for language is lacking, the larger brain size and cooperative behaviors implied by hunting, food sharing, and long-distance migration suggest more advanced communication abilities. Group cooperation would have been essential for hunting larger prey, defending territory, and raising young. The extended childhood implied by slower development rates in H. erectus compared to earlier hominins would have required greater parental investment and social support. The relationship between H. habilis and H. erectus is still debated. Some researchers view them as distinct species with a direct ancestor-descendant relationship, while others argue that they may have overlapped for a time, possibly representing different populations of early Homo. Regardless, the combined legacy of these species is clear. H. habilis initiated a shift toward increased brain size and reliance on tools, while H. erectus took those developments to a higher level, pairing them with new physical and behavioral adaptations that allowed for global dispersal. By around 300,000 to 140,000 years ago, H. erectus populations had disappeared, likely replaced by more derived species such as Homo heidelbergensis, which in turn gave rise to Neanderthals, Denisovans, and modern humans. Yet the achievements of H. erectus, long-distance travel, advanced tool-making, and fire control, set the foundation for all subsequent human evolution. In essence, Homo habilis represents the beginnings of human ingenuity, using simple tools to expand dietary options and adapt to changing environments. Homo erectus represents the next great leap, combining physical endurance, cognitive advances, and cultural innovations to thrive across continents. Together, they illustrate the gradual but profound transformations that carried our lineage from relatively small brain tool users to world-spanning, fire-wielding hunter-gatherers, paving the way for the eventual emergence of Homo sapiens. The emergence of Homo sapiens represents the culmination of a long evolutionary journey, during which hominins gradually developed the anatomical structures, cognitive abilities, and cultural behaviors that define modern humanity. Appearing in Africa around 300,000 years ago, our species evolved from earlier forms such as Homo heidelbergensis or a closely related ancestor, inheriting a legacy of bipedalism, tool use, and social complexity, while introducing distinctive physical and mental traits that set us apart from both our predecessors and our contemporaries, including the Neanderthals and Denisovans. Anatomically, Homo sapiens exhibit several key differences from earlier human species. One of the most striking is the shape of the skull. Instead of the elongated, low-vaulted crania of Homo erectus or Neanderthals, our species has a high, rounded skull with a vertical forehead. This change reflects the expansion of the frontal lobes, associated with higher-order thinking, planning, and problem-solving. The cranial capacity of Homo sapiens averages around 1,350 cubic centimeters, slightly less than that of Neanderthals, but organized differently to support more efficient neural connectivity. The face is relatively small and flat, with reduced brow ridges, and the jaws are lighter and less projecting, orthognathic, than in earlier species. Smaller jaws and teeth are likely linked to changes in diet, food preparation, and cooking, which reduce the need for heavy chewing muscles. The postcranial skeleton of Homo sapiens is generally more gracile compared to the robust frames of Neanderthals. Bones are thinner and less dense, and the body is proportioned for endurance running and efficient heat regulation, with relatively long limbs and a narrow trunk. This lighter build may have been advantageous in the warmer African environments where our species first evolved. However, it did not prevent Homo sapiens from adapting to a variety of climates once they began dispersing globally. Cognitively, the leap from earlier humans to Homo sapiens was profound. While previous hominins had demonstrated intelligence and social cooperation, our species shows evidence of advanced symbolic thinking, abstract reasoning, and complex language. Archaeological finds reveal early examples of symbolic behavior dating back over 100,000 years, including the use of pigments for body decoration, engraved patterns on ochre, and beads made from shells. These artifacts suggest an ability to represent concepts, convey identity, and engage in symbolic communication, capacities that underpin art, religion, and culture. Language, although impossible to reconstruct directly in its earliest forms, is believed to have reached a level of complexity in Homo sapiens unmatched by other hominins. The development of syntactic language allowed for the transmission of detailed knowledge across generations, the coordination of large group activities, and the sharing of abstract ideas. This greatly enhanced cooperative hunting, technological innovation, and social organization. The neural and anatomical adaptations supporting language, such as changes in the larynx, hyoid bone, and brain regions like Broca's and Wernicke's areas, likely evolved in tandem with cultural complexity. The comparison between Homo sapiens and Neanderthals is particularly instructive. Neanderthals, who lived across Europe and Western Asia from about 400,000 to 40,000 years ago, were highly intelligent, skilled toolmakers, and adept hunters. They had brains slightly larger on average than ours, but their cranial shape was different, with a more elongated brain case and less pronounced frontal lobe expansion. Neanderthals were also physically stronger and more robust, adaptations to colder climates. They made sophisticated tools, Mysterian technology, used fire, cared for the sick, and possibly engaged in symbolic practices such as burial of the dead. However, the archaeological record shows that symbolic expression and technological innovation appear earlier, more consistently, and more elaborately in Homo sapiens. Genetic evidence confirms that Homo sapiens and Neanderthals interbred, leaving traces of Neanderthal DNA in modern human populations outside Africa. These genetic exchanges may have provided adaptive advantages, such as immunity-related genes, but they also underscore that our species' emergence was not an isolated event. It occurred within a network of related hominin populations interacting and exchanging both genes and cultural traits. The spread of Homo sapiens beyond Africa, beginning around 60,000 to 70,000 years ago, is a testament to our adaptability. As we moved into new environments, we developed specialized tools, clothing, and shelters to survive in diverse habitats ranging from tropical forests to Arctic tundra. Innovations such as sewing needles, tailored garments, projectile weapons, and food storage techniques demonstrate both creativity and foresight. These advances, coupled with complex social networks, gave Homo sapiens a competitive edge over other hominins. Artistic expression flourished among early modern humans. Cave paintings, figurines, and musical instruments from sites such as Chauvet, Lascaux, and Whole Fells, dating back tens of thousands of years, display remarkable skill and imagination. These works are more than decoration. They represent shared stories, beliefs, and a symbolic worldview that reinforced group identity and cohesion. Another defining trait of Homo sapiens is the acceleration of cultural change. While earlier hominins experienced long periods with little alteration in their tool technologies, modern humans show rapid innovation and diversification of techniques. The Upper Paleolithic period, beginning around 50,000 years ago, saw a proliferation of specialized tools made from bone, antler, and ivory, as well as regional styles and artifacts, suggesting complex cultural traditions. The ecological impact of Homo sapiens was also unprecedented. Our species became highly effective at hunting large game, contributing, along with climate changes, to the extinction of some megafaunal species. We also manipulated environments through controlled burning, plant cultivation, and the domestication of animals, setting the stage for the later agricultural revolution. Socially, early Homo sapiens likely lived in groups bound by kinship, mutual cooperation, and shared cultural norms. These groups may have engaged in alliances and trade with others, exchanging goods and ideas across considerable distances. Such interactions would have promoted genetic diversity and the spread of innovations. The complexity of these social structures required advanced cognitive skills for negotiation, conflict resolution, and long-term planning. The eventual disappearance of Neanderthals and Denisovans, and the dominance of Homo sapiens, may have resulted from a combination of factors: competitive advantage in resource acquisition, greater resilience in the face of climate fluctuations, and possibly higher rates of cultural innovation. However, rather than a sudden replacement, the process was gradual and involved periods of coexistence and interbreeding. By around 12,000 years ago, Homo sapiens had colonized nearly every habitable continent, from Africa and Eurasia to Australia and the Americas. This global distribution, coupled with the diversity of languages, customs, and technologies, illustrates both the unity of our species and its adaptability to local conditions. The defining characteristics of Homo sapiens: anatomical modernity, complex cognition, symbolic culture, and cumulative technological change, enabled us to become the only surviving members of the genus Homo. The emergence of Homo sapiens was not a singular event, but the outcome of countless adaptations, innovations, and interactions over hundreds of thousands of years. By combining biological evolution with cultural evolution, our species created a dynamic feedback loop in which each advance in cognition or technology opened new possibilities for survival and expression. This synergy ultimately shaped the modern human condition, a species capable of imagining the future, reflecting on the past, and transforming the world in ways that no other life form has achieved. Cultural evolution represents a defining aspect of Homo sapiens, marking a transition from purely biological adaptation to the creation and transmission of knowledge, behaviors, and symbols across generations. While biological evolution shaped the physical and cognitive framework of humans, cultural evolution allowed our species to manipulate environments, coordinate complex social structures, and transmit cumulative knowledge in ways unmatched by any other organism. This process began with early symbolic behavior, likely emerging tens of thousands of years ago, and accelerated as language, art, religion, and social institutions became increasingly sophisticated. The development of spoken language was arguably the most transformative milestone in cultural evolution. While early hominins may have had basic vocalizations and gestures, modern human language enabled precise communication, abstraction, and the articulation of hypothetical scenarios. Language allowed humans to share information about food sources, predators, and social alliances, dramatically improving survival prospects. Beyond practical communication, language facilitated storytelling, moral instruction, and the creation of shared myths. Oral traditions transmitted cultural knowledge across generations, embedding lessons about the natural world, social norms, and spiritual beliefs. This capacity to encode and preserve complex information contributed to the formation of collective memory, social cohesion, and cumulative culture. Art represents another critical dimension of cultural evolution. Early humans created visual representations through cave paintings, carvings, and figurines, often depicting animals, humans, and symbolic motifs. These works, found at sites such as Lascaux, Chauvet, and Blombos Cave, illustrate the capacity for abstract thinking, symbolic expression, and aesthetic sensibility. Art served multiple functions. It reinforced group identity, documented environmental knowledge, and facilitated ritual or religious practices. Decorative items, such as beads and pendants, suggest early concepts of personal and social identity, status, and beauty. Art was not merely decorative, but a medium through which humans conceptualized and communicated abstract ideas, reinforcing cultural norms and values. Religious and spiritual practices emerged as early humans sought to explain natural phenomena, mortality, and the forces governing their environment. Burial rituals, often accompanied by grave goods, indicate beliefs in an afterlife and the importance of social memory. These practices provided frameworks for ethical behavior, social cohesion, and cooperation. Shared rituals and spiritual beliefs strengthened group identity and enabled coordination among larger social networks, facilitating resource sharing, joint hunting, and defense against rivals. The symbolic and ritualistic aspects of religion demonstrate the interplay between cognition, sociality, and culture, showing that early humans were capable of abstract thought that transcended immediate survival needs. Toolmaking and technology also contributed to cultural evolution by creating cumulative knowledge. While earlier hominins used basic stone tools, Homo sapiens developed increasingly specialized implements, incorporating diverse materials such as bone, antler, and ivory. Tools were not only practical but also culturally embedded, as knowledge of crafting and usage had to be transmitted through teaching and imitation. This transmission of technical knowledge exemplifies cumulative cultural evolution. Each generation could build upon the innovations of the previous one, leading to increasingly complex technologies over time. Such advances improved food acquisition, defense, clothing, and shelter, directly influencing survival and reproductive success. Social norms and governance emerged as another facet of cultural evolution. As human groups grew in size and complexity, informal rules and formalized customs regulated behavior, reduced conflict, and promoted cooperation. Shared norms regarding resource allocation, mating, and leadership created stable social structures. These norms were communicated through storytelling, ritual, and language, embedding ethical and practical knowledge into the culture. Over time, these social frameworks evolved into more formalized institutions, such as councils, chieftaincies, and eventually states, illustrating the coevolution of cultural, social, and political complexity. Cultural evolution also influenced cognition. The demands of social learning, language, and technological innovation selected for enhanced memory, problem-solving, and abstract reasoning. Humans became capable of planning for the distant future, simulating outcomes, and reflecting on past experiences. Cultural practices such as teaching, apprenticeship, and mentorship allowed skills and knowledge to accumulate far more rapidly than genetic evolution alone could achieve. The interaction between cognitive capacity and cultural complexity created a feedback loop. As culture grew more intricate, cognitive demands increased, which in turn facilitated further cultural elaboration. Trade and intergroup interaction further accelerated cultural evolution. As early humans expanded across diverse environments, they exchanged goods, ideas, and technologies, facilitating the diffusion of innovations across vast regions. This exchange promoted cultural diversity while also allowing the integration of beneficial practices, enhancing survival and adaptability. The spread of agricultural techniques, tool types, and symbolic motifs demonstrates how cultural evolution enabled rapid adaptation to changing environmental and social conditions. The role of cumulative knowledge cannot be overstated. Unlike biological evolution, which relies on slow genetic change, cultural evolution operates through learning, imitation, and teaching. This allows for rapid dissemination and refinement of adaptive strategies. For example, innovations in hunting techniques, food storage, clothing, and housing could spread across populations within a few generations, dramatically improving resilience and fitness. This capacity for rapid adaptation helped Homo sapiens thrive in diverse climates and ecological niches, from tropical forests to Arctic tundras. Language, art, religion, social norms, and technology are interconnected aspects of cultural evolution, each reinforcing and amplifying the others. Language conveys knowledge and symbolism, art and ritual encode and transmit values, and technology enhances survival and efficiency. Together, these elements create complex cultural systems that define human societies. Unlike biological traits, cultural traits are flexible and can be modified, adopted, or abandoned based on social and environmental conditions, giving humans an unparalleled ability to adapt. The emergence of culture also redefined the concept of adaptation. Whereas natural selection acts on genetic traits, cultural selection allows humans to adapt behaviorally and cognitively to immediate challenges. Cultural practices can mitigate environmental pressures, create new niches, and influence reproductive success. For instance, agricultural innovations enabled sedentary lifestyles, leading to denser populations, social stratification, and urbanization, changes that would have been impossible through biological evolution alone. By fostering shared understanding, cooperation, and innovation, cultural evolution allowed Homo sapiens to extend their influence beyond natural limitations. It enabled the creation of complex societies, transmission of knowledge across generations, and mastery of diverse ecosystems. Cultural evolution thus represents a powerful mechanism by which humans shape both their social and physical environments, ultimately allowing our species to dominate the planet. In conclusion, cultural evolution is a defining feature of humanity, complementing and amplifying biological evolution. Through language, art, religion, technology, and social norms, humans create, transmit, and accumulate knowledge, enabling rapid adaptation to changing environments. This process not only enhances survival, but also fosters identity, cooperation, and creativity, forming the foundation for civilization. Unlike other species, Homo sapiens relies not solely on genetic inheritance, but on the dynamic, cumulative transmission of culture, a mechanism that continues to drive innovation, social transformation, and our ability to imagine and shape the future. The technological and social revolution, often epitomized by the Neolithic Revolution, represents one of the most transformative periods in human history, marking the transition from mobile hunter-gatherer societies to settled agricultural communities. This profound shift, beginning roughly 12,000 years ago, fundamentally altered the relationship between humans and their environment, reshaped social structures, and laid the foundations for the rise of complex civilizations. Unlike previous stages of human evolution, which were primarily biological, this revolution was driven by technological innovation, strategic resource management, and social reorganization, demonstrating the unique interplay between human ingenuity and cultural evolution. Agriculture was the cornerstone of the Neolithic Revolution. Early humans began to cultivate plants systematically, selecting species that were high yield, easy to store, and nutritionally valuable. Crops such as wheat, barley, rice, maize, and millet were domesticated in different regions of the world, reflecting both environmental opportunities and human ingenuity. The domestication of plants allowed populations to stabilize in one location, reducing the need for constant migration in search of food. Settled agriculture also led to surplus production, which enabled the support of larger communities, facilitated trade, and provided the means to store food for periods of scarcity. Surplus production was not only an economic advantage, but also a catalyst for social differentiation, as individuals or groups who controlled surplus resources gained influence over others. Animal domestication complemented the development of agriculture, providing a reliable source of protein, labor, and secondary products such as milk, wool, and hides. Dogs, goats, sheep, cattle, and pigs were among the first domesticated animals, offering both practical and symbolic value to early societies. Domesticated animals enhanced agricultural productivity by assisting with plowing, transportation, and fertilization. They also became integral to emerging social and religious practices, often symbolizing status, wealth, or spiritual significance. The interplay between plant cultivation and animal domestication created stable, productive, and increasingly complex subsistence systems, reducing vulnerability to environmental fluctuations and fostering population growth. Permanent settlements emerged as a natural consequence of agriculture and animal husbandry. Villages and towns were established near fertile land and reliable water sources, leading to the construction of durable dwellings, storage facilities, and communal spaces. The architectural innovations of early settlements, including defensive structures, granaries, and religious sites, reflect not only practical adaptation, but also the emergence of symbolic thought and social organization. Permanent habitation facilitated the development of kinship networks, communal decision-making, and labor specialization, creating the first forms of coordinated governance. Settled life also encouraged the accumulation of material possessions, which contributed to social differentiation and the emergence of distinct social classes. With permanent settlements and surplus resources came new social structures. Hierarchies began to form based on wealth, access to resources, and control over labor. Leaders or councils emerged to manage communal affairs, resolve disputes, and organize large-scale projects such as irrigation, storage, and defense. This social stratification introduced the concept of unequal power distribution, which became a defining characteristic of early civilizations. Gender roles often became more rigidly defined, with men typically assuming agricultural, construction, and defense roles, while women managed domestic tasks, food preparation, and childcare. However, the specifics of social roles varied widely across regions and cultures, influenced by ecological, economic, and cultural factors. Trade networks expanded significantly during this period, connecting distant communities and facilitating the exchange of goods, technologies, and ideas. Early trade involved essential commodities such as grain, livestock, pottery, textiles, and raw materials like obsidian, copper, and flint. Trade not only provided access to scarce resources, but also fostered cultural exchange, spreading agricultural techniques, artistic styles, and religious practices across regions. The growth of trade networks encouraged specialization, as communities could focus on producing particular goods or services in exchange for others, further enhancing efficiency and interdependence. These networks laid the groundwork for regional integration and the development of economic systems that would later underpin states and empires. Technological innovations during the Neolithic Revolution extended beyond agriculture and domestication. Pottery, for instance, allowed for the storage and transport of food and water, preserving surpluses and facilitating trade. Tools made from polished stone, bone, and later metals increased agricultural efficiency and construction capabilities. Irrigation systems and water management techniques enabled humans to cultivate land in previously marginal areas, boosting productivity and population density. The accumulation of technical knowledge was often passed down through apprenticeship, imitation, and communal teaching, reflecting the increasing role of cumulative cultural learning in human societies. Religion and ritual played a significant role in consolidating social cohesion during the technological and social revolution. Early temples, shrines, and sacred sites served as centers of community identity, social regulation, and spiritual expression. Religious leaders often held social authority, guiding moral behavior, mediating disputes, and organizing communal labor for ritual or construction projects. Religious symbolism was closely intertwined with agricultural cycles, fertility, and natural forces, reflecting the deep connection between human survival and environmental understanding. These practices reinforced shared values and norms, facilitating cooperation among increasingly complex and diverse populations. The emergence of political structures coincided with technological and social advancements. As settlements grew in size and complexity, informal leadership systems evolved into more formal governance mechanisms. Leaders, councils, or elite classes administered resource distribution, coordinated defense, and regulated social interactions. Legal codes and customary rules emerged to manage disputes, enforce social norms, and maintain order. These early political institutions reflect an understanding of collective action problems and the necessity of structured decision-making in large communities. Over time, these systems became more elaborate, eventually leading to the formation of city-states, kingdoms, and empires. Population growth was both a driver and a consequence of the Neolithic Revolution. Stable food supplies, improved shelter, and protection from environmental hazards increased survival rates and fertility. Higher population density, in turn, intensified the demand for innovation, resource management, and social coordination. Densely populated settlements also facilitated cultural transmission, allowing knowledge, skills, and customs to diffuse rapidly within and between communities. This dynamic feedback loop accelerated technological development, social complexity, and the cumulative expansion of human culture. Environmental transformation accompanied technological and social changes. Humans began to manipulate landscapes on an unprecedented scale, clearing forests for cultivation, redirecting rivers for irrigation, and selectively breeding plants and animals to suit their needs. While these modifications enhanced resource availability and productivity, they also introduced new ecological challenges, including soil depletion, deforestation, and the spread of zoonotic diseases. The ability to alter environments illustrates a defining feature of human societies: the capacity to reshape ecological systems to serve social, economic, and technological goals. The Neolithic Revolution also set the stage for intellectual and artistic developments. Increased stability and surplus allowed individuals to devote time to activities beyond immediate survival, such as craft specialization, art, and ritual. Pottery decoration, figurines, weaving, and symbolic carvings reflect both aesthetic sensibilities and the codification of cultural identity. Early writing systems, initially developed for record-keeping and trade, later evolved into tools for historical, religious, and scientific expression. These developments illustrate the interdependence of technological, social, and intellectual evolution, highlighting the holistic transformation of human societies. Urbanization and the emergence of complex civilizations were direct outcomes of the Neolithic Revolution. Settled agriculture enabled the concentration of populations in towns and cities, providing the critical mass necessary for political centralization, labor specialization, and large-scale construction projects. Urban centers became hubs of economic, religious, and cultural activity, facilitating the exchange of ideas and innovations across broader regions. The formation of complex societies involved integrated systems of governance, taxation, law, and social hierarchy, creating structures that would persist for millennia and form the foundation for historical civilizations. In conclusion, the technological and social revolution represents a pivotal stage in human evolution, where cultural innovation reshaped subsistence, social organization, and human-environment interactions. Agriculture, animal domestication, and permanent settlements provided the basis for population growth, surplus production, and technological development. These advancements fostered the emergence of social hierarchies, trade networks, political institutions, and complex civilizations. By fundamentally transforming human life, This revolution demonstrates the profound interplay between cultural knowledge, technological innovation, and social organization, marking the beginning of humanity's capacity to shape both society and the environment on an unprecedented scale. The conclusion of the evolutionary journey, spanning from the emergence of the first living cells to the formation of complex human societies, provides a comprehensive reflection on the remarkable transformations that have defined life on Earth. The story of evolution is not simply a chronicle of biological change. It is a narrative of innovation, adaptation, and the intricate interplay between organisms and their environments. Understanding this trajectory allows us to recognize both the continuity and diversity of life, to appreciate the intricate processes that have shaped our species, and to gain insight into the underlying mechanisms that continue to influence the world today. Life began with the simplest of forms, single-celled prokaryotes capable of metabolism, replication, and adaptation. These first cells, though microscopic and seemingly insignificant, laid the foundational blueprint for all subsequent organisms. The emergence of cellular membranes, nucleic acids, and metabolic networks enabled the replication of genetic information and the harnessing of energy from the environment, which were critical prerequisites for survival and evolution. Prokaryotic life, diverse and adaptable, thrived in a variety of extreme conditions, demonstrating the remarkable flexibility inherent in living systems. The accumulation of genetic variation through mutation and horizontal gene transfer ensured the continued experimentation that would eventually give rise to more complex life forms. The transition to eukaryotic cells marked a profound step in evolutionary history. The development of a nucleus, organelles, and compartmentalized metabolic processes allowed organisms to increase in complexity, size, and functional specialization. The endosymbiotic acquisition of mitochondria and chloroplasts exemplifies how cooperation and symbiosis can drive evolutionary innovation. These cellular advancements facilitated the rise of multicellularity, enabling organisms to exploit new ecological niches and develop specialized tissues and organs. Multicellularity introduced novel strategies for survival, such as division of labor, internal communication, and coordination of cellular activity, which collectively enhanced the adaptability and resilience of organisms. The Cambrian explosion represents a striking episode in the evolutionary narrative, illustrating the rapid diversification of life forms and the emergence of complex body plans. During this period, the development of exoskeletons, sensory organs, and novel modes of locomotion expanded the ecological possibilities for animals, establishing the foundations for future evolutionary trajectories. The colonization of land further exemplifies the capacity of life to innovate in response to environmental challenges. Plants developed roots, vascular tissues, and protective cuticles, while animals evolved lungs, limbs, and mechanisms to conserve water, enabling both groups to exploit terrestrial habitats successfully. Vertebrate evolution highlights the emergence of key anatomical and physiological innovations, including the backbone, jaws, and thermoregulation. The progression from fish to amphibians, reptiles, and mammals illustrates how incremental modifications can lead to significant adaptive advantages, allowing organisms to inhabit diverse environments. The emergence of primates, with their grasping hands, binocular vision, and expanded brain capacity, set the stage for the evolution of hominids and ultimately Homo sapiens. Early hominids, such as Australopithecus, represent transitional forms that combined arboreal and terrestrial adaptations, bipedal locomotion, and social behaviors, reflecting the complex interplay between environmental pressures and evolutionary responses. The genus Homo, including Homo habilis and Homo erectus, illustrates the increasing importance of tool use, social cooperation, and cognitive development in human evolution. Homo habilis' ability to fashion stone tools and exploit diverse food resources expanded ecological possibilities, while Homo erectus' mastery of fire, long-distance migrations, and complex social organization demonstrate the growing role of culture and technology in shaping survival strategies. The emergence of Homo sapiens marks the culmination of these trends, characterized by advanced cognitive abilities, abstract thought, language, and symbolic behavior. Anatomical adaptations such as a high forehead, rounded skull, and smaller jaw reflect both functional and cognitive evolution, distinguishing Homo sapiens from other hominids. Cultural evolution, emerging alongside biological evolution, has become a dominant force in shaping human existence. The development of spoken language, art, religion, and shared myths facilitated complex social interactions, cooperation, and the transmission of knowledge across generations. Cultural practices enabled humans to manipulate their environments in unprecedented ways, fostering innovation, social cohesion, and collective problem-solving. Technological and social revolutions, beginning with the Neolithic Revolution, illustrate how cultural knowledge can drive ecological and societal transformation. Agriculture, animal domestication, permanent settlements, trade networks, and early political structures reflect the interdependence of technology, social organization, and environmental management. Throughout this evolutionary journey, several overarching themes emerge. One is the centrality of adaptation, both biological and cultural, in responding to environmental challenges. Genetic variation, natural selection, and phenotypic plasticity have enabled life to endure and thrive under changing conditions. Similarly, cultural innovation, ranging from tool-making to the development of social norms, has allowed humans to transcend immediate environmental constraints, creating new possibilities for survival and growth. Another theme is the role of cooperation and symbiosis, evident in the formation of multicellular organisms, endosymbiotic relationships, and social networks, highlighting the evolutionary advantages of collaboration over competition alone. Understanding the evolutionary history of life and humans offers profound insights into our present and future. By tracing the continuity of biological processes, we gain perspective on our physiological and genetic heritage, recognizing how traits inherited from ancestral species continue to influence health, behavior, and adaptation. Examining the emergence of culture and technology illuminates the mechanisms by which humans shape both natural and social environments, providing lessons in sustainability, innovation, and resilience. The evolutionary perspective also fosters an appreciation for biodiversity, ecological interdependence, and the consequences of human activity on global ecosystems. Moreover, studying evolution underscores the contingency and creativity inherent in life. The path from single cells to complex human societies was neither linear nor predetermined. It reflects a series of experiments in adaptation, survival, and innovation. Random genetic mutations, environmental fluctuations, and cultural developments all played interacting roles in shaping the trajectory of life. This awareness can inspire humility and responsibility, reminding humanity that our current achievements and challenges are deeply embedded in a long continuum of evolutionary processes. In conclusion, the journey from the first living cells to modern human societies encapsulates the dynamic interplay of biology, environment, and culture. It demonstrates the capacity of life to innovate, adapt, and create ever more complex forms, culminating in a species capable of reshaping its surroundings, transmitting knowledge, and reflecting upon its own origins. By understanding this history, we gain not only a scientific perspective on our existence, but also a framework for addressing contemporary challenges, from ecological stewardship to social organization. Appreciating our evolutionary past enriches our understanding of the present and informs our vision for the future, emphasizing the profound interconnectedness of life and the enduring legacy of evolutionary change. Good night.